An overview of circadian rhythms, and then I'm going to be talking about the two problems we're going to be tackling today. The first one is detecting periodic molecular species, and the second one is, given the measurement of multiple molecular species, can we tell the time this measurement was taken? So circadian rhythms are very fundamental for biology. They're seen in everything from fungi to plants to mice and humans. As we know, with plants, they'll open during the day and close during the night. And with mice, if you were to monitor their activity, you'll probably get a graph something like this, in which they're really active during the night and then inactive during the day. And these patterns happen with a period of around 24 hours. Circadian rhythms are also very fundamental for medicine. Disruptions in the circadian rhythm are linked to a lot more than just jet lag. They're also linked to depression, obesity, premature aging, and even cancer. So with the plants that I mentioned before, how they open during the day and close during the night, people wondered what would happen if you put these plants in a dark room with no access to sunlight. And even though they were in a dark room with no external cue telling it when to open and close, people noticed they still opened and closed at around the same time. So that led them to come to the conclusion that there was probably some internal clock inside the plant telling it when to open and close. Now, thanks to high throughput technology, we can now analyze organisms on the microscopic scale. We can analyze things such as transcripts, metabolites, and proteins. And as it turns out, these also oscillate with a period of around 24 hours. So we have the macroscopic and the wake-sleep cycle, which has a period of 24 hours, and then now we have the microscopic, which you also found out often when they're oscillating have periods of 24 hours. So this is the autonomous core clock. This is the key component that drives circadian rhythms in cells. So what we have here is a classic negative transcription-translation feedback loop. So clock and BMAL bind to the promoters of genes in cells, and they promote the transcription of these genes. In particular, they bind to the genes in the cry and purr family, which get transcribed and then translated into proteins. These cry and purr proteins combine to form a complex, which then bind to the promoters of clock and BMAL, inhibiting their production, which then inhibits the transcription of other genes in the cell. This happens with a period of around 24 hours, and this is a classic negative transcription-translation feedback loop. So the first problem we're going to tackle, identifying periodic molecular species. And if it is periodic, what is the period, phase, and amplitude? Now, there are a couple things that make this problem particularly hard. The first is that the data is very sparse. If we have data of around 24 hours, there's only about 5 to 7 samples across those 24 hours. In addition, there are very few replicates, so there will be about 1 to 3 replicates per time point, which makes estimating the true mean pretty hard, which actually makes the data very noisy. There are a few existing software packages that try to tackle this problem. The first one is JTK Cycle, which uses JT tests and Kendall's tau tests. The second one is ARSR, which uses spectral analysis. And the third one is LUM-SCARGLE, which uses periodograms. And then finally, we have Metacycle, which combines these methods into one method. Now let's take some examples of what identifying these periodic molecular species might look like. This is transcriptome data from mice. It's taken across 24 hours and it's sampled every 4 hours, giving us 6 time points. And then there's 3 replicates per time point. So let's look at these. Do you think these are periodic or aperiodic? And if they're periodic, what's the period, phase, and amplitude? Let's do the same for these. Periodic or aperiodic? What about these? Do you think these are periodic or aperiodic? Now, some of you might think some are obviously periodic and others are obviously aperiodic. However, all of these signals were labeled as periodic by at least one of the existing software packages. So it was apparent to us that there is a need for better methods. So we first started out with Fourier methods. Now, these performed well on synthetic data when the data wasn't sparse. However, as the data became more sparse in order to mimic real-world data, it started to perform poorly. So we tried Gaussian processes. You can see here we have a kernel for modeling periodic signals and a kernel for modeling aperiodic signals. Now, this performed better than Fourier methods. And finally, we tried deep learning. And deep learning is just neural networks with many hidden layers. And this performed the best out of all the methods that we tried. Now, in order to train our deep learning method, we need training data. So as it turns out, we can actually train our deep learning method using only synthetic data. So we can create periodic signals using combinations of sines and cosines, and we can create aperiodic signals from just random white noise or very simple formulas to create aperiodic signals. And since this data is synthetic, we have as much training data as we need to train our algorithms. And our data set consists of millions and millions of examples of periodic and aperiodic signals. Now, previous papers that have analyzed methods on this problem were really only able to quantify these methods using synthetic data. However, we created the first labeled real-world data. So these are signals from transcriptome experiments in mice, and we labeled both periodic and aperiodic signals. Now, the way we did this is to label periodic signals, we looked at the core clock genes. These genes have been shown to oscillate across a variety of different conditions. And we put them through human inspection, and if they looked periodic, we labeled them as periodic. Now for aperiodic signals, we use the three previous methods I told you about, JTK cycle, Arthur, and Lum-Scargill. If they all said that this signal was aperiodic with a p-value of 0.95 or higher, we put them through human inspection. If they looked aperiodic, we labeled them as aperiodic. So our deep learning solution is called Biocycle. It determines whether or not a signal is periodic. If it's periodic, it determines the period, phase, and amplitude, and it uses synthetic data to train. So this is a feed-forward neural network. There are actually two of them. There's one for doing binary classification, periodic or aperiodic, and this uses a sigmoidal output. And the second one is used to estimate the period, and this has a linear output. Now, once we have estimated the period, we can then estimate the phase based on the period, and once we have the period and the phase, we can estimate the amplitude based on that. So here are the results for binary classification for periodic and aperiodic signals. On the right here, we have AUC, and on the left here, we have ROC curves. You see on the synthetic data, Biocycle, or the deep neural network method, or as we shorten DNN, performs better than all the other previous methods. And for the real-world data, it's comparable to all the other methods. And we'd also like to note that only the previous methods were used to label aperiodic data. Biocycle wasn't involved in labeling the aperiodic data, but it still performs comparable to all the other previous methods. This is the results for estimating the period, phase, and amplitude. We can see Biocycle performs the best when estimating the period and the phase, and it's comparable to other methods when estimating the amplitude. Now, the second problem we want to tackle is what time is it? Oftentimes, there are experiments that are carried out at single time points. For example, say we have a mice with a control condition and then mice with an experimental condition. So it might be put on a high-fat diet, or it might be given some kind of drug. When the gene expression in the control condition is measured, and then the gene expression in the experimental condition is measured, people do differential analysis to look at the difference between the expression levels in these genes. However, these measurements are often taken at different times. So if we have a difference in the expression level, say in the experimental condition, in a certain gene, the expression level is lower, you might be tempted to come to the conclusion that the expression level inhibits the production of this gene. However, if we take circadian rhythms into account, it might just happen that due to circadian rhythms, the expression level in this gene is lower during this time of the day anyway. So we want to be able to impute the time measurements were taken in an experiment so that we can account for the confounding factors due to circadian rhythms. So the data we have is from gene transcript experiments in mice. We have data from a variety of different tissues, including tissues in the liver, tissues in the kidney, and tissues in the brain. We have 360 training examples and 147 testing examples. Now, for each experiment, we get about 20,000 genes. However, since we only have 360 training examples, it's hard to learn the relationship between these 20,000 genes and what time it is. So we shrink this down to 16 core clock genes, and this is the input to our deep learning method. And we got all of this data from the Circadiomics website right here. So, as I said, given the measurement of multiple molecular species at a single time point, can we tell what time those measurements were taken? So our deep learning solution is called Bioclock. It imputes the time measurements in an experiment were carried out, and it uses a specialized unit I'm going to be talking about that regularizes the model and gave us better results. So this is our deep learning architecture. The output actually mimics a cosine and sine on this 2D circular manifold. Now this 2D circular manifold mimics a 24-hour clock. The reason we use this is because we wanted to capture the cyclic nature of time. We tried using a linear output, however, 0 is very far away from 23 hours. Intuitively, 23 hours should be right next to 0 hours. We also tried using a softmax output, but this still didn't capture this relationship. So what we did is we forced the output to lie on this 2D 24-hour circular manifold, thus capturing the cyclic relationship of time. Here are our results. We had a mean absolute error within 1.2 hours for wild-type conditions and 2.6 hours for experimental conditions. We then used this architecture to impute the times at which geo-experiments were carried out. And we would also like to note that these error rates are well within the standard sampling rate for a lot of circadian experiments, which is about 4 hours. We also wondered what would happen if you trained a deep neural network on tissues from the liver and then tried to take that network and tested it on tissues from the brain. What we found was that if you trained on tissues from one organ and then tried to test on tissues from another organ, you got very poor results. So these specialized networks that were specialized to a certain tissue did not generalize well to other tissues in the body, in different organs. So we got the best results from training on all the different tissues. Now, we want to leverage unlabeled data. There's very little labeled data, but there's a lot of unlabeled data that we can use. So if we have this autoencoder, which projects this data on this 2D circular manifold that I talked about before, we could leverage this unlabeled data. We've been trying to do this for about a year. We're improving this right now. What we're trying to do right now is actually initialize this first half of the autoencoder using supervised learning. Therefore, we overcome this phase problem, where in the circle does 0 lie. And then we want to fine-tune it using the plethora of unlabeled data that we have. So in summary, I talked about circadian rhythms, why they were important for biology and medicine, and then we talked about how we can use deep learning for determining whether or not a signal is periodic or aperiodic, and we can also use deep learning to determine when measurements were taken in an experiment. So I want to end with a potential application for this. This is the list of the top 100 FDA-approved drugs, and with these drugs, they target genes that have been known to oscillate. So let's look at diabetes right here. These genes have been known to oscillate in organs such as the heart and the liver. If we knew when the expression of these genes was the highest, the doctor could know when to tell the patient, the patient should actually take the drug. And this could reduce side effects, this could make drugs more effective, and it could reduce the amount of drugs you need to take to get better. Thank you.
Thank you for a very interesting talk. Are there any questions? Please come to the microphone.
Well,